Okay, so we're going to have just a quick session today on how to make a homology model and like where do you go to get PDBs for a homology model. So the place that I go is this place called the PDB, the Protein Data Bank. So that's at www.rcsb.org. And you come here and you can search the PDB archive for any PDB that you want. But before you can, so sometimes you come into a project and your professor will give you a PDB, or you know, you might know proteins, you can put a protein name in here, like I could put in hemoglobin, and it will pull up lots and lots of PDBs that you can try to use. But what if there are no structures and you have to start from scratch? So there are websites out there that will do sequence alignments for you. There are also programs in these like both CCP4 and the Phoenix suite, which will basically take sequence alignments that you make elsewhere and then use them to make a homology model. So in this example, all right, I am going to start over here. There is a place, and I put this into the links, it's called HHPred. And you come to HHPred, which is at this white website over in DE, so I think it's in Germany. It has a whole bunch of different homology searches that it does for you, and I use the one called HHPred. A lot of these multiple sequence alignments work in a sort of similar way, and I use this one because I like to use Phoenix.mr Rosetta and then make Rosetta build as much of my structure for me as possible. So this is one of my personal ones that I like to use. And so what you give this one is you give it a sequence. So for example, I can give it a sequence that I'm going to copy and paste into it from a file that a collaborator sent me. And okay, so I just had to take out that little caret, else I thought it was empty. And you don't have to give it the star because that's the end. And so here's a sequence right there. And you can select which like PDB version it's running down here. So I'm just going to use the default. I don't want to use these other ones, but these are different databases down here that you can run homology searches on. Whoops, I unselected that. And then you can also select different type of proteomes if you want to use it. I am not going to select a proteome. And then I give it a custom job, and I can call this K class. And then you hit submit and it runs. Okay, so it's not going to run it again because I submitted this an hour ago to test this. And so it's actually K-IRA right here. I'm just going to click it. It's going to give you the results right here. And it makes this cute little visualization. It says, I took your sequence and I found all these different PDBs that it matches up to either in whole or in part. So like this one matches just this part, might be a domain of it. This structure here matches the whole thing. And I did a different one. This is like a heterodimer that I was doing for a collaborator. Whoops, right here. And so you see this one, it found two that matched up a lot. And then these littler sequences right here, it didn't match up as much. And so then it takes the sequences. I'm looking at this. So it has all these, so you can tell people have solved a lot of ones that match this domain up here. Lots and lots. And then it comes down here and it tells you what they are. So it finds, this is the PDB ID, and it finds a match for chain A in that PDB ID. It says, I think that this is 99.9% probably what this is. And it assigns it an E value, which is basically how good it is. So you can see that this one is, even though the probabilities are very close here, that it's more likely to be this PDB than this PDB, because the E value is like super small. It's negative to the 10 to the negative 25th, and this is 10 to the negative 15th. These are all very similar already. But if you look at it, so like if you can see here, the number of aligned columns. So my sequence that I gave it was 337 amino acids. This one aligned 327 amino acids. Whereas down here, even though this still says it's 99.5% the same, it only aligned 148 amino acids, only 148 columns. And it just goes on down, does every single hit that it showed you here in pretty colors. And it goes on for many pages, you can see like that. And then down here is the actual alignments one by one. So here is the first one, 60D1 chain A. And this is the SS underscore pred is the secondary structure prediction. This QK IRA is my sequence that I gave it. Next is what it says is the consensus to the 60D1. And then it matches, and then it says these amino acids here don't match. This amino acid doesn't match. This is amino. So it shows you all the ones where it doesn't match. And it gives you like these H's are, it thinks it's helical. You know, like if it would have charged amino acids. And it goes on down, and it shows you like how it predicts. My sequence would overlay the secondary structure prediction for 60D1A. And then it does all the ones in the predictions, and you can see as it goes on, like this one has a lot of matches in it. You know, it's just a few that don't match. And then you have more. So now I could either go to the PDB, and I could put 60D1 into my PDB search. There's many ways to do this depending on how automated you want to be. And the PDB will give you a whole bunch. I can download the sequence for this. I can download the PDB itself. You know, I could just right click and save it. And then I can take this PDB after I've saved it, and I can go to Phoenix, and I can say, I want to run SCULPTOR. And I can give it the PDB. Where did I put this one? And then I can give it a sequence file. And I could just have it basically sculpt it to see how it would work out. So it would just take that, and it would, so I could take the sequence, and I could just try to overlay that sequence on this PDB, you know, based on sequence alignment, and just see how it goes. I can give it a sequence alignment. SCULPTOR can try to do its own sequence alignment. On the command line, and I know that I'm torturing all of you guys with this thing about a command line. I need to give you another command line shell. There is a way to use what's called a multiple sequence alignment in the HHPred format. You just give it as a keyword file name. So the way, so here it looks like the way you would do it is you would give it a multiple sequence alignment, which I have right here. And let me open up that file and then give it a target index. So when it comes down, I have to double check how that file actually looks. It doesn't run.
Let's click on the magnifying lens next to the browse button.
This one?
No, on the sequence alignment. Your other sequence alignment window. Yeah.
Oh, okay.
You had one open.
Oh, here we go. All right. Gonna pop it up.
Should
Only if I have like 1,000 of these windows open now. Why won't something open? Because I already have a window open. See, this is one of the things before is I couldn't make it open up these things to edit them and look at them. See, it's just not.
Yeah.
Because I thought it would let me if I just. All right, let me just.
Yeah.
So the way you get it, you can go to HHPred and it says download HHR. And you can just click that and download it. And let me, I like how this is like, oh, I have so many windows open. It's not even funny how many open windows I have open. I have them everywhere on God's green earth. Okay, so when you open up the HHR file, it tells you its query, what it was trying to do, the command it sent, that's the command it sent to the web server to run this. And then here's the same scores that I talked about, like right here. It's just all summarized at the very top. And you can see that they're each assigned a number 1, 2, 3, 4, 5. So you can, if you want it to like basically do, I don't know how to say, like all of them, then you basically, you can say you want it to do in the command line, you, what it should be is that it's, I am suffering from too many windows. I've lost my window. So it says target index 1. I could tell it target index 2, and it should pick the second one. I could tell it target index 3, it would pick the third one out of that huge long list. Or what you can do in the command line when you're still trying to figure out why the GUI is not reading your file correctly is you can actually go in here and I either delete or I comment all of these out. So you can actually go through. You can comment them out. You can like say, I don't love you. I only want one, which is this first one right here. And then you can just delete everything else. Which I'm not going to do right here because this file is like very, very long because there are lots and lots of hits. Because it gave me 733 out of 3,200 hits in this file. So it's just really long. And I'm not using Emacs to do this, which means there's not a one click, which means I'd have to like highlight and drag all the way to the end of this. But there is this lovely, lovely website here. Model editing with SCULPTOR. And you can basically, when you give it an alignment, it will, in SCULPTOR on the command line, you can basically say, I want you to use SCULPTOR, and I want you to, I don't want you to just do the first one. I want you to do the first 10 and give me 10 SCULPTOR outputs of all 10 of them. And I'm going to try all 10 of them in my phaser. Or what you can just do is you can just give it, you could go into SCULPTOR and just say, I don't care about the multiple sequence alignment. I just want you to do it with that sequence file that I gave you. So let's delete one of these. Should do it. I give the sequence file. I want to apply it to chain ID A. And let's see. You have one additional configuration file open. All right, I can't run it because I have too many windows open and I don't know where they are. But you can do many files at once. Some people use this thing called AlphaFold. So you can go in and you can find folds and domains using sequences and models to use, that it will go through and edit your stuff. There are many ways to do this, but the big places that you want to go with these are, I have so many CCP for Windows open, are, that's web browser tabs, is the PDB, the protein data bank, right, where you can download the PDBs that you want to modify. And you want to then use a program like SCULPTOR to modify it. And for those things, you will need either, you need the sequence of your protein of interest, and you generally need an alignment. But again, like as I said, you can actually have, SCULPTOR just give it its best guess, which I have had it do before. All right, so another question. So Catherine asked if SCULPTOR only worked with HHPred. No, so SCULPTOR will also work with .ali alignment files. I think those are the ones out of SwissProt. You may have to edit them a little bit, because so if you have so a bunch of the, so here we go. It'll use Clustal right here, format, pure format, and ALI. But I think the ones out of SwissProt are .alis. So that is, so if you don't get a sequence, if you don't have a PDB to start off your Phoenix search for running a molecular replacement, this is how you would make one.